Medical research is a subject which many patients are keen to better understand. They're interested to know what is happening to find a cure or better treatments there are for sarcoidosis. Now, I'm delighted that Dr. Katie Bechman is here to talk about current and future sarcoidosis research. Katie is clinical lecturer in rheumatology and inflammation biology at King's College London. Katie.
So thank you. Thank you for that introduction. So my name's Katie. I'm a clinical lecturer and a rheumatology doctor based at King's College London. And I'm going to talk today about the future of research in sarcoidosis. I want to thank Sarcoidosis UK for inviting me to talk today. So I put this photo up. This is Dr. Jonathan Hutchinson, and he made the first ever known description of sarcoidosis nearly 150 years ago. And so I think that's a really good time now to look back and really consider what we understand about sarcoid and what we yet don't know and where research is heading. And I'd like to talk a little bit about that today with you all. So my first question about where research is heading is really looking into what causes sarcoidosis. So what do we know already? Well, we know it's a combination of how your genes and environment interact. We know that sarcoidosis is more common in clusters of families. You have an increased risk if your sibling does, but we haven't actually identified one specific gene that would be responsible for sarcoidosis. And it's important at this point to consider that sarcoid really is an umbrella term for this huge variety of different clinical presentations. And sometimes we have to unpick that to be able to study it better in our research. So what do we know? Move this out of the way. What do we know about genetic research at the moment, and where does it stand? Well, traditionally, genetic studies look at a candidate gene. So we examine a gene that is of interest to us, and we can compare that between individuals with sarcoid and healthy individuals without sarcoid. But to look at a candidate gene, we really have to already understand the pathology of sarcoidosis. Now, over the last few years, we've had a revolutionary in human genetic research with the use of genome-wide association studies. This allows us to look across the whole of an individual's genetic makeup. We can compare individuals with sarcoid to those that don't, and we look at small changes in DNA between these two groups of people. And this allows us to identify a far greater number of genes. So what have we identified so far in sarcoid? Well, we found some genes that increase the risk of you developing sarcoid. And we've also found certain genes that associate with different clinical presentations. And what we are now seeing with some research is that we can identify some of these genes that are producing proteins that can then be measured in the blood. And you can see that there would be clinical applications for that in the future. So I've included this slide, which I know is very busy, but it's really just to give you an idea about all the different genes that have been identified that associate with sarcoidosis. So each one of these lines is a chromosome. Chromosomes have lots of DNA within them, and each of those DNA will code for a certain gene. And as you can see, these are all the different genes that have been identified as associated with an increased risk of sarcoid. So we're definitely making some progress there. So what is the future of genetic research in sarcoidosis? I've just put this on for referencing, but we have had huge advances in our technology in genetic research. We have next generation sequencing, which means that we can sequence and look across a huge amount of DNA over a short period of time. We're also able to use multi-omics. So that allows us to study not just DNA, but proteins and molecules that are produced by the body. We're now starting to see research into how the genetics and the environment interact with each other. And that is the initial trigger for sarcoidosis. But as I've already said, one of the things that is evolving is that we really need to examine patients with sarcoidosis who share similar physical traits. So we know that people have lots of different clinical presentations. And if we look at individuals who have similar clinical presentations, it allows us to have much clearer and accurate results. And additionally, we need to be looking across diverse populations, across different ethnic groups, as we know people with different ethnicities can present differently with sarcoidosis. So my second question, and something that I'm really interested in, is how common is sarcoidosis? Now, as Professor Hart has already alluded to, sarcoidosis affects the whole world, but is more common in certain areas. So much more common in the Northern Hemisphere, as you can see on this map here, in Canada and Scandinavia, have much higher rates. The rate is around 1 to 15 new cases per every 100,000 people. And as you can see, looking here at the UK on the map, we were estimated to have a number of new cases between 1 to 5 per 100,000 people. Now that data now is 25 years old, and it was also based on only about 2% of GP practices. So here at King's College, we've really been trying to look into this more and find out how common sarcoidosis is in the UK. And we found it actually is much more common than previously thought. So the number of new diagnoses is around 8 per 100,000. But interestingly, you can see that the number of new diagnoses has increased over time from 2003 all the way to now, with a very large increase happening around 2011. And this probably relates to the fact that we're better at diagnosing sarcoidosis. We now have new imaging techniques that help us diagnose it better, and we have better techniques for being able to get a biopsy sample to look for granulomas, which is the hallmark of the disease. But you can also see from this graph that there are changes in the number of males and females who have the condition. So it used to be quite equal split, but now we're seeing that there's a greater number of men with new diagnoses of sarcoidosis. And in my next slide, you can see that there's also been changes in the age range. So we do see sarcoidosis more common in individuals between the age of 40 to 60. But from 2011 onwards, as you can see with this blue line, there's an increase in the number of new diagnoses in individuals who are over the age of 60. And again, this may relate to the fact that we're better at diagnosing this condition, which may have been labeled as something else in the past. So my third question is, how can we better diagnose or monitor the disease? And what is the research that's looking into this area? Well, as you may be aware, we don't have one single blood test that tells us if an individual has sarcoidosis or not. We do have some markers that are used in clinical practice. So you may have heard of the serum ACE, which we sometimes measure and is elevated in some people with sarcoidosis and goes up with disease activity in some people, and may also help us know if someone's going to have a relapse of their sarcoid. But there are some new promising biomarkers on the horizon. Now, the second one in that table, soluble IL-2 receptor, is being used in some clinical practices across the world. And this is also, like the serum ACE, a blood test that goes up in individuals who have sarcoid and also goes up with disease activity. However, it goes up with other things too. And so we need to be careful about how we use these biomarkers. There is lots of research looking at promising new markers that go up better with disease activity, and also some biomarkers that help us work out an individual's prognosis. And if you look on the bottom of the column, this KL6, there's been some research looking into the fact that rises in that biomarker level can associate with more scarring in the lung. And you can see how this may be clinically applicable in the future if we're able to use these biomarkers to help guide treatment options for patients. So that's blood biomarkers, and now thinking more about imaging of the disease. And some of you may be aware that in clinical practice, we can use an imaging modality called a CT PET scan. So this is a CT scan of the body where an injection is given with a small amount of tracer that's taken up into the body and taken up by active immune cells involved in sarcoidosis. And as you can see in this image here, where we have a head all the way to the thighs, this black area here is the lungs. And as you can see, there's some increased uptake in the lung area that shouldn't be there, which is demonstrating active sarcoid immune cells in the lungs. And the next image along, which is slightly translucent, you can see the dark areas are areas where the tracer has been taken up, which is occurring in the joints, in the knees, in the elbow, but also there are some lymph nodes under the armpit showing tracer uptake. Now, CT PET scans are really helpful for us in identifying disease burden, in identifying areas of the body that we didn't know to be involved with sarcoid, and also in identifying areas that we could maybe target for a biopsy. But they do have some disadvantages, that they're costly, they take time to perform, and also they're associated with radiation dose. And so we don't want to be doing these scans too frequently. So what can we maybe do instead, and what is in the current research? Well, we're looking at new imaging techniques. And so I've just included one today, which is a total body PET. Now, this is still in research and isn't available in clinical practice, but this is similar to the standard PET, but instead of just doing a small amount of the body, it takes an image from the head all the way down to the toes. Because of the technology, it provides a much more detailed picture of the entire body. It's very quick to do, lasting only a couple of minutes compared to around an hour for a standard PET scanner. And it's associated with a much lower radiation dose. And so you can see this would be very helpful in clinical practice when we're trying to monitor disease activity. And I've just included in this slide just how detailed these pictures are from the total body PET-CT, where you can literally see every single element of the human body. So my next question is thinking, who should we treat with sarcoidosis? And I know Professor Hart's already alluded to the fact that there are a number of patients who have the disease that resolves without needing any treatment whatsoever. But unfortunately, there are a proportion of patients who do require treatment, and some who have quite severe disease that require multiple different treatments. And so trying to understand how to tease apart these two groups would be extremely helpful for clinical practice. And so this takes me on to talk about personalized medicine. And this is really what we should be aiming for in the future in medicine. This is a way, instead of using a single treatment in a one-size-fits-all manner, where some people will respond, others won't, and others will have side effects, that we're able to use some diagnostic techniques, which may be genetics, it may be a biomarker, where we're able to see which treatments work best for which people and give them the correct treatment that they're most likely to respond to and that they're less likely to have side effects from. Now, we're not yet there with sarcoidosis, but we're starting to see some evolving research that tells us that we can predict certain things in terms of disease course. So as I've already alluded to, some of our genetics studies are able to identify individuals who are more likely to have an acute presentation that's self-resolved, and genetic tests that can identify patients who are more likely to have a more chronic and burdenous disease. We've also looked at some studies that look at the immune cells from biopsy samples and looks at the type of cell within that biopsy. And there are certain cells that associate more with a self-resolving disease, and there are other cells that associate with more chronic disease. And lastly, looking at clinical predictors, and there have been studies that have identified the organ that you present with may help guide the course of your disease. And understanding this in more detail may help us in the future be able to guide treatment and give more reassurance to patients. So my fifth and final question, which I think probably is one of the most important, is what are the treatment options and where does the research stand looking at different treatment options for sarcoidosis? So as you may or may not be aware, not all individuals need treatment, but those who do, the first line treatment is often steroids. In some individuals, we'll start a steroid-sparing treatment, such as methotrexate. But if patients do not respond to steroids or steroid-sparing treatments, we often have to put them on a biologic drug, or if they have a very severe disease at presentation. And we're lucky in England, we now, since 2023, have infliximab, which is an anti-TNF. It's a biological drug that's approved for the use in refractory disease. So this slide here is slightly complicated, but I've really just included it to just highlight all the different possible treatment targets along the different stages that the immune system plays in contributing to sarcoidosis. And so if we go through each of these, we have medications that may be able to target the initial trigger, that could target that first immune cell activation, that could target the messenger chemical proteins, these cytokines. And as you can see in here, we have TNF, which is the medication that infliximab targets, medications that can target granuloma formation, medications that can help increase these regulatory immune cells that regulate this immune response, and also some medications that target scarring in the lungs. And so there have been loads of research looking at different medications that target each one of these areas. So I thought when we're talking about medication in sarcoidosis and what the research has shown us up to date, I think it's quite important just to put a timeline on. And this is looking specifically at immunosuppressant medications for sarcoid. And this timeline really shows us how things have changed in the last 20 years. So if we just look firstly at infliximab, which is the anti-TNF medication that's licensed for use in sarcoid, we had several studies from 2006 onwards. We then had some trials looking at other anti-TNF medications. And then we had some studies looking at medications that are used for arthritis. And they had now been looked at to see if they work for sarcoid. And this is where the landscape kind of stood for some time. And then if you look from 2021 onwards, we have 5 new trials dedicated for sarcoid. So this has been a huge expanse in the last 3 years. I think it's a really exciting place we're in at the moment, that sarcoidosis is receiving attention from the research community and from pharmaceutical companies. So when we are considering trials in sarcoid, I think it's important to also just remind everyone that not every trial is successful, and we haven't had success throughout the whole journey. So I've color-coded these as a traffic light system, but the green ones indicate trials that have been successful. So our infliximab trials and the sister TNF drug adalimumab were very successful. We've had more recently trials looking at new medications for sarcoidosis that have also been successful. But it hasn't always been like this. There have been certain trials that haven't really made any difference. And there have been some trials that have been associated with worsening of sarcoid, and no further trials in these medications have been continued. So where do we stand now at the moment? Well, I went onto the clinicaltrials.gov website, which is a website where trials, especially for interventional medications, are registered. And we currently have 69 ongoing future or completed trials in sarcoidosis, which is a huge number and a massive difference from where we were, 15 years ago. And these trials are looking at treatments that are already used with established drugs in other conditions, which is the story with infliximab, or it's looking at new drug molecules specifically targeting sarcoidosis. And I thought it'd be helpful to go through the 4 trials that have occurred in the UK in the last year. So Resolve Lung was so I should first start by saying all of these trials are a trial that has a placebo arm. So you get either put into the active treatment arm or you get put into the placebo arm. So we can monitor patients who haven't had treatment, so we get an idea of how effective the actual active drug is. So Resolve Lung was a trial that was taking place in individuals who had lung involvement in sarcoidosis. And it looked at namilumab, which is a GM-CSF inhibitor. This is one of those key messengers that's involved in driving granuloma formation and inflammation. This medication was given as a subcutaneous injection, so under the skin, and you were initially given it for 6 months, and you were either in the active treatment arm or the placebo arm. And after 6 months, everyone in the trial was then able to receive the active treatment. Now, global recruitment has now shut for Resolve Lung, but we should be hearing some results from this treatment in the near future. The next trial is EZOFIT. This is efzofitimod, which is a neuropilin-2. It binds to neuropilin-2 on the immune cells to reduce inflammation. And this is a medication that is given once monthly over a 12-month period, and you are either in the active treatment arm or the placebo arm. And global recruitment is also shut for this trial, and again, we should be hearing results in the near future. So the next 2 trials are trials where recruitment is ongoing. So the first is XTMAB16. This is a newly developed TNF inhibitor, which is blocking the same messenger protein that infliximab blocks. And this is given as an injection over a 12-week period. Either you'll be getting the active drug or the placebo. And afterwards, everyone is eligible to enter into an open label period where they will receive the drug. And this is for individuals with lung involvement, but also for those who have lung involvement and other organ involvement too. And to be eligible for this, you would have to have had active disease despite being on steroids or another immunosuppressant therapy. And the last one is the KITE trial. This looks at an OD01, which is a chitinase inhibitor. So this blocks any change of cells from to make them pro-inflammatory or make them pro-fibrotic, so trying to cause scarring. And this is actually a tablet. It's given over a 12-week period, again, either in the treatment arm or the placebo arm. And there's no open label period for this trial. But importantly, you don't need to be on steroid or immunosuppressant medication to be eligible. You just have to have active sarcoidosis. So I thought I'd end this trial part of the presentation by talking about how patients can get involved in trials. So just first to say that there are varying entry criteria for current trials or trials that are newly being developed. They've generally been for pulmonary sarcoid, but I hope this is going to change. And we've got some trials in the States that have been carried out in individuals with cutaneous disease. They tend to exclude individuals with severe disease, anyone with heart or nerve involvement. And you should have evidence of active disease on a scan that helps you become eligible for the study. You don't always have to be on immunosuppressant medications. As I said, XT-MAB, you do need to be on steroids or an immunosuppressant, but you don't have this requirement for entry into the KITE study. Most of the trials have an open label period, which means that if you're put into the placebo arm, at the end of the study period, you have the ability to then receive the drug for some time. But there is some commitment on your behalf, and it would require in-person visits. This is often every 2 to 4 weeks, and sometimes with a telephone call during it. I should say there are huge amounts of benefits to getting involved in research for the medical community, but also for yourself. And I would advise that if anyone is interested, the best thing to do is to speak to your clinical department to see if there's any locally available studies, or to check out the SarcoidosisUK link with the clinical trial data on it. So I hope in this presentation I've tried to give you some updates into what's happened in research and what is happening now in terms of looking into what causes sarcoid, how common it is, how we can improve our ability to diagnose and monitor the disease, thinking about who we should be treating, and what are the updates in treatment options. I'll take any questions now, if there are any.
Thank you very much, Katie. That was fascinating. I have to say, as a non-clinician, I was really concerned this would sail over my head, but thank you for speaking my language as well.
No problem.
We have loads of questions. You've covered a couple of them. So I think one that probably interested me first is around genes. And one of the questions is, do we carry a certain gene, or is there any research surrounding causes? And then could this gene be identified and then screened?
Yeah, so I think the genetic research in sarcoid is really interesting. And as I said in the presentation, there's not one sole gene that we've identified that causes sarcoid, and it's really a mix of lots of different genes working together. But we're getting much better at identifying what those genes may be, and with advances in our genetic technology, I think that's only going to improve. And I think that the goal really is that in the future we'd be able to identify a gene that we can then test in the blood very easily that may help us guide whether a person has a diagnosis of sarcoid, or to help guide what the disease course may be. And I think that's where we're heading, and we're just not there yet.
Okay, but is there research also looking at how we improve and speed up the diagnosis process?
Yeah, so I think this is really important, and we've made huge progress in the last 10, 15 years, and I think that it is illustrated on the fact that we're diagnosing a lot more patients with sarcoidosis who may not have been diagnosed in the past. We're definitely improving in our ability with improvements in scanning. We have improved abilities to biopsy, for example, doing endobronchial biopsies. And so I think we're improving our ability to diagnose, and I think that's going to continue with the advent of new scans. I think we're definitely making progress, and I hope that continues.
Okay. I think one of the big buzzwords all around the world at the moment is AI, artificial intelligence. Where does that sit today with research into sarcoidosis?
Yeah, so I think, within medical research, AI is making a huge impact. I think for sarcoidosis, there are certain products at the moment that are using AI specifically within genetic studies, but also looking at imaging and seeing whether we can better diagnose sarcoidosis on, for example, PET imaging by using AI modalities. So it is being incorporated in different elements of sarcoidosis research, and I'm hoping that's going to help improve things in the future.
Okay. I think you talked about the 4 major clinical trials that are underway. Are there any trials available for newly diagnosed patients as well? Or is the expectation someone's had sarcoidosis for some time, therefore they're well established with their own symptoms?
Yeah, so I think the important thing to mention at that point is that there are a number of patients with sarcoid who the disease will resolve spontaneously. And what we don't want to be doing is giving someone an unnecessary treatment that they don't need. We also don't want to be recruiting that patient into a trial because that would muddy the results. We may think the drug is better than it actually is, whilst really what was happening is the disease was resolving by itself. So I think one of the issues with sarcoidosis trials is making sure that we're choosing the right patients to go into the trials. It's quite a heterogeneous disease. People have loads of different clinical presentation, and that sometimes can muddy the waters. So I think it's really important about choosing the right patients for the trials and choosing the right outcomes to look for, because different people who have different organ involvement will have different outcome measures that we want to look at. In terms of an individual with newly diagnosed disease, I think if it's someone who we think needs immunosuppressive treatment, for example, a steroid or something stronger than that, then they would be eligible for research. And as I mentioned, the KITE study is recruiting people in that kind of stage of disease who aren't necessarily yet on a treatment, but would be someone who we would be putting on one if they weren't going into a trial. And I think things will continue along that trajectory. I think there'll be more trials looking to see if there are better drugs than steroids that we can be using for our patients. So I think that's definitely, on the horizon, but we just need to be 100% certain that when we design clinical trials in sarcoid, that we're really careful about choosing the right patients and designing these trials with the right outcome measures.
Well, in fact, you touched on my next question, in fact, there around the steroids. The question we've had here is, what other medication options are there for sarcoidosis apart from steroids? And is there research into alternative options? Now, you mentioned one in your trials page there, but just tell us a bit more about that, perhaps.
Yeah, so for lots of patients, we know steroids work very well. For some individuals, we would start them on a steroid-sparing agent, which is what we do in clinical practice. And there have been trials looking at different steroid-sparing agents, seeing which ones work better for which clinical presentations. And there are studies currently now looking at whether going on a steroid-sparing agent from the beginning is more helpful than maybe starting with a steroid and seeing how things go. So there are trials looking in that initial phase of starting a steroid. And as I mentioned with the KITE study, that would be a new interventional medication that's not been used before, and seeing how well that works. So I think in that kind of initial period, there are trials going on, but a lot of the trials I discussed today were looking at individuals who aren't responding to that initial steroid with a steroid-sparing agent, and those that are needing more stronger treatment. And there's lots of trials looking into that. So I think we're definitely far, in a much better position now than we were 5 to 10 years ago. And I'm hoping that things are just going to continue along this way.
Okay. Your final question. We've talked about the various types of sarcoidosis, and lung clearly comes up as a major side of it. One question that's been asked is, though, is any research looking specifically at lung damage arising from sarcoidosis rather than general lung damage, which we know has been researched for many years?
Yeah, so there are. So I should first say that most of the trials are looking at lung sarcoidosis. It's the most common organ affected by sarcoid, and so a lot of the trials looking at the immunosuppressants and the medications that modulate the immune system are targeted at lung involvement. There have been some studies looking at targeting scarring of the lungs in sarcoid, and taking some of the knowledge we know from other scarred lung diseases and seeing whether similar treatments work for those, the antifibrotic medications. And so there are lots of trials looking specifically in the lung in terms of immunosuppressants, but also in terms of and anti-scarring medications. And so I'm hoping this is going to help kind of change the way that we treat individuals when we have, you know, more varied options that we can use. So that's the hope really from all this trial program.
Brilliant. Well, thank you very much for your time there. I think it's very exciting times. I'm looking forward to seeing what comes from these trial results later this year. They can't come too soon, I think, for all of us. So let's hope for positive output.